नहीं ऑन ही रहेगा और ये कैमरा तो है बनाया हुआ यहाँ इसकी ब्राइटनेस बहुत कम नहीं है
So we'll wait for a couple of minutes for others to join in, and maybe within 10 minutes time we can start with the class, right? Just one person has joined, and if it is below 5, I'm not going to continue. Because हमारे पास mail आया है कि we have a meeting with NPTEL on 31st, and we'll get back to you उस matter पे क्या बच्चे interact कर रहे हैं नहीं कर रहे हैं तो बता
क्लास थे उसके बच्चे थे
कौन सा चल प्रॉपर 20 कुछ
इसमें 4 ले
या
नहीं एक काम करें आपको चाय दे दे तब तक
अच्छा यार
ऑल द बेस्ट
रेडी लेंगे यार हाइट को भी मैच
लिखने में कंफर्टेबल अभी तो हाँ एक चीज बस स्टार्ट करने वाले को बता देना और दूसरा चाय आपको दे दी बीच में नहीं
हाँ
दे देना
दे देना So we'll wait for a few more minutes, right? So by 6:10, we'll start with whatever number of participants have joined in, right? Okay, so by the time we wait for some of you to join further, I'll start with my introduction. Is my voice clear to all? Just some of you can give me a confirmation. Am I properly audible to all of you?
Yes, ma'am, all clear.
Okay, thank you. Salva, right. So I think the idea is very clear. You all are receiving weekly updates on some of the course contents, right? So week wise, today's class is for the first week contents that you have already received. So all my classes is going to be on the all these live interaction session with me. It's going to be on the Tuesdays, 6 to 8 p.m. And for any doubts that you have had for the previous week's content, that we can take up during this session, right? So the idea why NPTEL has arranged for such live sessions is for you to get any doubts clear that you come across in the recorded lecture, and it's very obvious that you cannot. So there are discussion forums. I hope you have all got the links to the discussion forums where you can send in your written questions, and you obviously have some of your colleagues or the teachers or someone else answering those for you, right? So make sure that you join all such discussion forums online. And if there is something which is still not clear to you, or maybe you just want to go through, or maybe you just want to go through all the things, so we would begin our class. All the live interaction sessions will first begin with us giving you a brief overview of whatever has been discussed. We'll try to go slow so that all of you get the chance to engage in and ask your doubts, whatever is there, through the course of the class, right? So you need not wait for me to finish the entire class before you ask me a question. Also, I'll try to keep the class as far in English as far as possible, but I think it will always be nice if we can interact in a bilingual mode so that others who might not be very comfortable with it get the most benefit of it. Is that okay with everyone? Is there anyone who has a specific problem with this? So if there's someone who you feel like is not very comfortable in English and some important concepts are being discussed, so you can stop me in between and you can ask me to also explain them in a language that you might be comfortable in, right? So the idea is for all of you to benefit most of from this live interaction, and we will start from the very basics because it's also the discussion from your first week's course content. So it obviously has a overview. So overall, see, what is very important with immunology in general is you have to understand the subject with a lot of schematic which is flowing in a proper direction, right? If you just start picking out one topic from somewhere, you can understand that one topic in exclusivity, but for you to understand the subject as a whole, it is very important that you move systemically, right? So we will start with the basic definitions of immune system, who all are the players in it, and in like just like a summary of the overall course, right? For today's class, we will next move on to the history that is related to the immune system, some of the very important people who have laid the foundation to this subject, and then we begin learning about in detail components of the immune system again. So in detail, if I straight away jump with the details of say innate immunity, you might not very well understand the entire complexity of it today in itself, right? So we'll try to get in detail, but just enough for you to understand and be familiarized with all the terms that we are going to use throughout this course. And I'll not just uh, get into the very precise details of all the topics that we discussed, right? Okay, I think that is good for an introduction of how I'm going to proceed by. My classes, unless and we still have people joining. Okay. So unless and until we have a very important, some complex topic that is coming in, I personally would prefer attending lectures wherein we have the blackboard mode of teaching. And I think the other live interaction session that you might have already attended is already in a presentation mode, right? So it is my belief that while we have a blackboard teaching setup, the students get a chance to interact better, and to keep it a little different for the benefit of all, we have tried to do it this way. So as far as my class is concerned, we might not have PowerPoint presentations that I would show unless and until it is very important for you to better understand a topic. But if there's any change that you would want me to do while I'm taking up the class, that also you can let me know, right? So it's already 6:15. I think we can start with our course curriculum. If there's anything in between that you would want me to repeat, or any other thing related to the particular topic I'm covering, you can please stop me right over there. It's right there itself, and we will first take up your doubts and then we'll move ahead on to the next topics. Okay. So, to introduce myself, I am Debalina Chatterjee, and I am a PMRF fellow in the University of Delhi in Department of Zoology. Right. So my research is also related to immunology, definitely. So I'm working on the resident immune cells of the brain in context of neurodegeneration. So based on our respective research fortes, we can apply to be the teachers to different courses in NPTEL, and that's how I got a chance to interact with all of you. Okay. So So the course title Immunology. So as we must be understand have we must already have an understanding that logy is to study something, and over here it is the study of the immune system. So immune system, if we see that system in isolation, it is a system. It is a defense mechanism of the body. So it has evolved to protect us from the diverse microorganisms that surround us. Not all the microorganisms that surround us is therefore able to cause us a harms, right? So you must know that you must be knowing that your entire gut per se is densely populated with a lot of microorganisms, but none of them, the ones that are resident to our enteric system, they are not capable of causing us any damage or any disease, because there is something in our body that is protecting us from the harmful consequences of their being with us. So what is this system that is working to protect us? That is your immune system. So immune, the word immune is the state of protection. So it's a word that comes from Latin. Immune is for exempt, right? So immune system. So immune over here is a word that comes from Latin, and it means to be exempted. So exempted from what? From diseases. So it's a state of protection from diseases, right? Okay. So, one very interesting fact, and that's somewhere why I got interested in taking up immune system in context of neurodegeneration, is there are only two systems in the body. So there are only two systems in the human body which has a overlapping set of function. Overlapping as in, so we have certain features which is very common in the immune system and in the nervous system, right? So if you think of your nervous system, if you think of your brain per se, what do you think it does, right? When you're looking around, we are seeing, we are, when I say that we are seeing, it's the brain that is actually recognizing a lot of object in our environment, right? In our external surrounding. So if there is an object, say, which is of interest to us, we say we are getting attracted to it. So if there is an object that is of disgust, so we repel it, right? So first, my brain is recognizing it. It is then trying to put up a response And say if I go in a particular direction and I know that there is something good that might come if I walk towards it, it's the human tendency, it's a tendency of the human brain to remember the way towards something that is rewarding. Say it has a memory, right? So whenever you are seeing something in the surrounding, you are recognizing it, you are having a reaction to it, or you are having a response to it. And if that response you feel like it's that particular thing is of interest, and it might, this particular experience might be of use to you in the future, your brain also has a memory towards it. All these three features are there in your immune system. How, which part of the system, which cell, or how it is able to do all of this, is what we are going to understand in the entire course of our immunology, in the entire immunology course, right? So the overlapping feature between the nervous system and the immune system is its ability to, so so there is a point of equivalence between the immune system and the nervous system in terms of their ability to recognize, respond, and have a memory. Okay. Now, let's first start with recognize, right? What is it recognizing? In this entire surrounding that you must be sitting at, there's a lot of microorganisms, there's a lot of other things that can actually breach your state of protection and can enter the body, right? Whenever your immune system is coming in contact with any such microorganism, some of which might also be pathogenic, it is able to recognize them. So the immune system So the immune system has the ability to recognize all the microorganism, the entire diversity. So well, you must have studied microorganism, or a general knowledge about the amount of microbes that surround us. You must be able to appreciate the diversity, right? It's not like, so you might have a genus, a species, a strain of it, substrains of it. There's a lot of classification that keeps going on in the microbiological system. All these minute differences in all of these organisms is recognized by the immune system. Next, it has to respond to to all these organisms, and how it responds is something that we will understand in a great detail. So the major part of your immunology course is actually about the responses that the immune system mounts up once it recognizes your pathological or your microbial or any such foreign entity, right? Now, when I'm talking about foreign entity, it is very important that your immune system is only going to recognize and respond to a foreign entity. Say if it starts mounting a response against your own body cells, this protective state can also cause harm to the same individual. So that is a dysfunction of the immune system, and that is again very important for us to understand, because that also understates a lot of practical applications in the medical biology field, right? So the response and the recognition only has to be towards a foreign system. Only then we can say that it's a properly functional immune system. If I have to still classify this response as a brief summary, we have two responses. One is the effector response. Effector, as the name suggests. So if the writing is not clear somewhere, I hope that you are able to follow my words, right? So effector response, something is changing, is having an effect. It is doing something to change something, about the situation. It is responding in a manner to make an effect to clear off this to clear off the whatever pathogen it has now encountered. And memory in itself is the second way of response. So there are two responses that the immune system mounts. To immediately take care of the attack, it mounts a effector response, and then it also remembers this encounter in a memory response. So what is memory response? That whenever I am going to encounter this pathogen once again, I would know that how I tackled this challenge the last time. So the immune system this time do not have to think over again ki which pathogen is it. So when your immune cells are recognizing the pathogen, it is actually putting up a lot of effort in doing so, and that costs some time. In that time, the pathogen is able to cause some problems to you, because of which you might see some primary responses in your body, say fever, right? But when the next time the pathogen is again encounter, immune system from its last encounter remembers this. It now already recognizes this pathogen quickly, and it doesn't have it do not have to think that which of the effector responses I might have to give this time, because it already remembers that what worked the last time. So this memory response is nothing but a rapid and heightened. So last time, if we say that from the previous encounter, the effector response was, say, 2 times. So maybe two molecules of the effector, two units of the effector molecules was released in 20 minutes time. Now, the immune system can produce within 2 minutes 20 effector molecules. So it is rapid and the response is also heightened. Okay. So this is the basic overview of the immune system. Now to do all of this, what is exactly happening? How is the immune system bringing into effect all these responses? To better understand that, we will have to get into that what all is the components of the immune system. Because as I said, if I have to understand immune system as a whole in the first go in itself, it's a very complex network. It's multiple pathways playing its role at the same time, right? So we will we to understand that we have to first break up this entire system into certain components, just for the ease of our understanding. By the end of this course, we will actually bring in all these different components together, and we will see that how a infection is cleared off. So to give an overview before we move on to discussing the history, which in itself is very interesting, let's just have an outline of what all is the components of the immune system. So we break while we are reading about the immune system, the biggest classification that is made, the most broad classification is its division into innate and adaptive immunity. Just give me 1 second. Okay. So innate, right? As the name suggests, innate is something that you have since the time you were born. So you were, whatever features or whatever behavior that you might not have, that you didn't require to learn with the course of your life, the memory that has been passed on. Say, you can also say that it's a trait that you already knew by the virtue of your birth. So any such character that is there, it is known as innate. So people who might be having an animal behavior background, they might also know that there is something called as innate behavior. What is innate behavior? Just to give you an example of what this word means, say when you are born, no one would tell you that if you touch a hot object, you have to remove your hand off it, or any aversive stimulus, you have to repel it. No one actually teaches it to a toddler, right? Whenever they have a aversive situation, they automatically know that they might just have to move their hand off it. Or say, you can also say suckling, the baby suckling, that is also a very important example of innate behavior. No one teaches a newborn baby to do it, but it just knows by the virtue of its birth that it has to do that particular behavior in order to acquire nutrition, right? So such things, such behaviors which have not been taught through your course of life experience, those are known as innate behavior. And while we are talking in context of immune system, we are calling it the innate immune system. Okay. Adaptive immune system. Adaptive, again, in biology it will be very easy for you to understand any component or any topic if you just try making the literal meaning of the words that you encounter. Adaptive, so you're adapting to something. So through the course of life experiences, you're adapting through your experiences and you are learning something, right? So it's a learned response. So if I talk about adaptive immunity, what comes in is a response that you give, the memory response, say, because one component of memory is that you have had an experience and then now you are responding to it. So memory that we were talking about so far is actually a feature true to the adaptive immune system, and it's not in the innate immune system. So if I talk in terms of the necessity for both these players, it's we can never say that one part is more important than the other. We'll understand why I say so as we follow through. But to say, people say that innate immune system is the more primitive one. So primitive is not because of the functions that it performs. It's not like it is performing primitive functions. Primitive because that is present since the origin of life on earth. So every organism that you see, they have certain innate defense mechanisms to it. That's even true to the bacteria, which knows to protect itself from maybe a more dominant harmful bacteria or microorganism in its surrounding by producing certain antimicrobial agents. So you must know that say the penicillin, it was isolated from a bacteria which was showing, because of its ability to clear off the nearby growth of any other microorganism. So that synthesis of that particular antimicrobial peptide is an innate defense system which is even there in the smallest bacteria, right? So innate immunity is present in everyone. That's why it is known as primitive, because it is present even in the primitive phyla, right? So it is present in both vertebrate, invertebrate, microorganism, any that you can identify. In adaptive immune system, this is a more advanced one. Again, advanced because it is only present in the vertebrates. We do not find adaptive immune system in the lower phyla, right? Okay. So if I say innate, so we'll come to innate and adaptive maybe in today's class itself. But just to say that this one has no memory and this has no specificity. Innate immune system has no memory, no specificity. What do I mean by specificity? That this innate system is going to respond to every microorganism in the same manner. So not like if it encounters a fungi, it is going to put up some different mechanism, and not that if it would encounter some other similar fungi, it will have a different response, right? So it has no specificity. But yes, we will learn that innate system is able to distinguish based on certain molecular markers. It can distinguish between a fungi and a bacteria. There are certain molecular signatures which is unique to every class of microorganism, and your innate immune system is able to recognize them so that it can accordingly mount a response. But say bacteria, will it respond differently to E. coli and Staphylococcus? No, it's not specific. Over here, we see that there is memory and there is also specificity. So as we were discussing, it will not only have a specific response to E. coli and Staphylococcus, it will also have a very different response to different strains of E. coli. So it's very, very specific, right? Okay. So, Swarangi, we have already summarized our discussion of the overall overview of the immune system, and we'll move on to the history part of it. So far as there is anything that you would want to ask, you can please ask. So it's your first class. We'll try and go a little slow, but so I'll keep on asking you to ask me questions, and I'm not sure how new you are to the course or what is the educational background that you are coming from. So people who are coming from biology background, I'm expecting them to be more familiar with these things because they must have studied about it in their pre-graduation courses, assuming that you're all graduate students. Okay, if there's no questions, we'll move ahead. So we will get into the history part of the branch, which I particularly find very interesting because actually there were a lot of stories that led to the development of this branch in general. Right? I'll just keep bulleting some points, and for reference, so this is the book that all my lectures is going to be, QB. You can follow any edition of this, right? Professor Ghosh and Professor Ganguly, they have also referred to this book along with some additional books for their particular lectures, but most of my content is going to be from QB, right? So everything that I say and talk about, you're going to find that back in this particular textbook. So how the field of immunology developed. The first observation of something able to protect someone, right? That was made by, so there was this Thucydides, right? So he was accounting. So it was during the it was during the time of war, wherein he saw that the soldiers who had actually recovered from plague, and only they were able to nurse someone who is currently suffering from plague, right? So that is the first account of a state of protection that ever that is ever documented, right? So there must be casual observations that people might have made, but no one actually documented these things. So the first record is very, very old. It's during the 430 BC, right? So it is recorded in a statement that those soldiers who have recovered from plague are the only ones who could nurse the sick, as they would not contract the disease a second time. After that, 2000 years later, this observation came into medical practice. So it was not like how vaccination is in medical practice now. How it started, we had the Chinese and Turks. So what were they doing? They were injecting smallpox pustules through the cuts in skin. So what they would do, they would purposely create some cuts in the skin, and they would try to brush off these smallpox pustules on these cuts, right? And they would then see that if I give a weak if I make a weak culture of the smallpox and I brush it off on through the skin cuts, people who have gone through this procedure, they would not contract the smallpox disease. Again, in smallpox, we had Edward Jenner, right? Edward Jenner made another observation that milkmaids who were essentially working in the dairy farms, so milkmaids who were exposed to cowpox. So cowpox is the causative agent belongs from a similar family. So this people who have been infected with cowpox, if they are now so this is first exposure. When they're for the second time exposed to smallpox, there was no disease. Again, so what was happening here, that they would purposely try to inject the smallpox, say the lice, whatever. So if someone is infected from smallpox, they would try to take some fluid portion and try to rub it through the breaches in skin. And over here, so it was, so what was happening in this is that someone who has been infected, their body has already fought off that smallpox infection, and that's why that particular fluidy secretion only had weak cultures of smallpox. But over here, milkmaids who were exposed to cowpox, just because cowpox belongs from a similar family of pathogen, but it is not as efficient in causing a disease, the milkmaids who were exposed to cowpox did not contract this disease then. When they now encounter smallpox, they would not be falling sick. Because what is actually happening, now that we have had a brief overview, that the body must have had a memory of a similar pathogen coming in, and when it encounters it, it knows it pretty much knows how to respond to it, and might be that's why they were not falling sick. So it's not might be, that's what we know right now, but these are the observations that build the basis of the of this immunology discipline, right? The next key observation was made by Louis Pasteur. So if you remember the stories, it was not going to be very difficult for you to memorize the names, right? So that's why it is I feel it's a way to remember these names, because there are many competitive exams which are going to ask you these things, the names of scientists. As trivial it might seem for you to remember these names, but these things can, so one or two questions here and there, that can cost you a lot sometimes, right? So it is important for you to remember these things. So what Louis Pasteur saw, Louis Pasteur's case was of chicken and cholera. He was working with Vibrio cholerae, and he was able to isolate he made it possible to isolate pure cultures of Vibrio cholera, and he was trying to understand the mechanism using the chicken model. Now, how it is said in story is that he went on some breaks, and when he came back, he used some old culture that he was having to inject it into the chicken, and he actually he was expecting the chickens to fall sick, and then he would continue on his study. But to his surprise, these chicken did not develop any disease. So the chicken which was injected with some old culture of cholera, it could not cause any disease in the chicken this time. So he thought that it might be because the culture has gone bad. So what he did, he grew some fresh culture, and because the supply, so there are uh, two limitations that actually led to such an important discovery. First, that he injected a old culture of this cholera, and next, when he had a fresh culture, his supply of chicken was limited. So he was forced to carry out on his studies using this old chicken stock that he had, which he had already administered with the weak culture. Once he put in his fresh cholera in this previously injected chicken stock, what he observed is that again the chicken did not die. So he thought that there is something that is definitely happening. Either the chickens have developed some super interesting mutation which is altogether protecting it from the infection by cholera, but that was very less likely since mutations of such great significance do not just happen in a day. It needs to go through multiple generations, right? So he thought that he tried to perform the same experiment in the similar manner in using cholera and chicken. So he would always use some old culture of bacteria, put it in the chicken, and then administer the same chicken with with fresh cultures, and the chicken would always live. He also did the same thing with your sheep and using the anthrax bacillus. So same thing. Bacillus anthracis, it would cause a disease, but weak administration in sheep, and then that particular sheep again giving a strong culture, it did not die. So he here understood that it is not because of any miracle or any mutation. There is something called attenuation of the strain which is happening. So attenuation is nothing but weakening the pathogenicity of a microorganism. When it is getting attenuated, so it is not causing disease, right? So this concept, he developed it into the concept of vaccination. So this is where we are still heavily dependent on. So, this science actually came up from one mistake and one limited supply of chicken, right? So to try it on further, he again administered a 8-year-old boy. So from the birds to the mammals, where he used sheep, he has finally come on the humans, wherein an 8-year-old boy was given rabies vaccine. So rabies vaccine was the first to be tested by Louis Pasteur. This 8-year-old boy was given a rabies vaccine. So therein the vaccine was nothing but just an attenuated culture of rabies virus, and purposefully in some following years he was bitten by a rabid dog, and this boy survived. So that was the first time vaccination happened on the humans, and it was the foundation of immunology. It forms the foundation of immune protection till date, right? So there's a course of vaccination that every little baby has to undergo now so that it is protected in the future. Okay, so Pasteur did everything. It understood that vaccination worked. It understood that attenuation can be used to develop vaccine. The only thing that he failed to understand is how this vaccination is working, right? So it wanted to he did not understand that how exactly this mechanism is working, but he nonetheless is given a very huge credit. But what followed was even more interesting, because now we are getting an answer to how it's working, right? So it was an experiment by it's Emil von Boring and Shibasaburo Kitasatu. Okay. So in 1890, In 1890, they were actually given a Nobel Prize for the same concept that we are going to discuss. So what they saw was that when they isolate, so now the concept of vaccination is there. So the concept of immunization is there. Someone who has received the vaccine is now called immunized, right? So when what they did, they would isolate the serum component. Do you all understand the difference between serum and blood? What is the serum component that I'm talking about? So a blood which is devoid of the clotting factors, it is now called as the serum component. Okay. The serum component is what actually has all the nutritive components that might be present in the blood. It's just that it doesn't have the I'm not sure. I might have to check, but I think it doesn't have the RBC components too, if I'm not wrong. Right? So you guys can check that, and you guys can tell me. You guys can also see it through during the class. Just trying to interact as much as possible, right? If any one of you knows that, you can also just unmute yourself and you can ask, and you can let me know, right? So that I can also learn through the class. Okay, so they isolated the serum component from people who either received vaccination or who have previously suffered the disease, but it was not having fatal outcomes. And when they put the serum in a non-immunized individual, this individual was, so when the pathogen is now encountered by this non-immunized individual, there is no disease. So it understood that there is something in the serum component which, if I take out from an immunized individual and I put it in a non-immunized individual, this individual is also protected from the disease coming up, right? So he did not know what to name it. So he just said that the serum might be having certain anti-components. So what anti? So they just named it antitoxins, something to precipitate out whatever is causing the disease. So it like a quencher, or say like a magnet. So it works to take care of whatever is there to cause the disease, and it so that's something. Agglutination is something that is related to it. Still have people joining, right? Or there can be something called as precipitin. So what precipitin? It would settle down any microorganism that was able to previously cause the disease. So these are certain names that different researchers gave to whatever is present in the serum, until the time we got to know that what is exactly there in the serum. So something called as the immunoglobulins that we will come to came into into the knowledge of people much later. But first, von Behring and Shibasaburo Kitasu got identified this particular feature. After this, we had two school of thoughts coming up, right? One was by Elvin Kabat, and Elie Mechanoff. So you have to understand the time period that we are right now referring to, right? So this is in the 1800s, right? So at that time, it was still the technological advance allowed us to play around with blood samples. So the people could get blood from humans, and they would try and do different possible experiments from it. But people from biological background, and if some of you might have sometime heard, you can write now there are facilities for us to grow different cells in artificial culture conditions, right? So cell culture was not there previously. So Elvin Kerbath, he proposed the humoral model of immunity. So what is humoral? Humoral has got to do something with the fluids. So he proposed humoral immunity. What he saw is there is a particular fraction. So Bering and Kitasato, they said that's serum, but now he says that there is a fraction in the serum. There is a fraction in the serum that is now known as gamma globulins or immunoglobulins. Only that particular fraction, if that is transferred to non-immunized individuals, they also attain a state of protection. So gamma globulins, we now know that what is there in it. So everyone, I'm sure, must have heard of the word antibody, even if you are not at all from the biological background. Let's thank the pandemic for it. So we now know that what is there in that serum is nothing but the antibodies, right? So this is in 1800s again. But before this humoral immunity came into the limelight, there was a scientist who gave a pretty advanced proposal for his time, which we now know is equally important, which is cell-mediated immunity. So as I was saying, that since the cell culture techniques were not very well developed at that particular time, cell-mediated immunity, the discovery of it, happened much later. It gained attention rather much later because Mechanov actually proposed it before Kabat, but it failed to receive attention because people could not understand that what is there. So what was exactly happening in cell-mediated immunity? Apart from serum, Mechanov saw that if he would transfer certain cells, so which cells the WBCs, the white blood cells, which we now know is the main component of the main cellular component of the immune system. We will discuss all the types of these WBCs in detail in the following classes, right? If it was seen that when certain cells, the WBCs, were transferred from one individual to another, it could confer the state of immunity to this non-immunized individual. And he also say that these cells, which cells in WBCs, as I was saying, there is a huge classification of WBCs which is possible based on its function, based on the lineage of its coming up. So the differentiation during the development course, the lineage of it. I'm sure if you have attended all the lectures of Professor Ghosh and Professor Ganguly, you must have encountered that particular flow diagram where he discusses all the different cell lineages. I, for today's class, will not get into it because I believe that you must first have a very clear picture of the overview, then only we'll get into all the details, and with the differentiation we will also discuss the function of each of those cells. So we'll keep that for the next week. But for today's class, if you have to understand which particular WBC he identified, he then did not know, but all he knew was these WBCs was showing phagocytosis, right? So now if we see, it must be, so there are only two WBCs which have phagocytic capacity, which is the monocytes and the neutrophils. So likely Mechanov identified the monocytes and neutrophils as responsible for cell-mediated immunity. It was much later with advancement in the, so scientists across the world got divided into two groups. Majorly they were in favor of humoral immunity, while there were some who were trying to work through the cell-mediated component of immunity, building upon Mechanov's idea. But much later, this debate was settled when people got to know that it is very possible that both these components are very, very important and both are equally responsible. Both is equally important and equally responsible for transfer of the immune state, right? So humoral immunity is somewhat similar simpler to understand, because there's just one thing that is there, which is antibody. Not just one, but it's more simpler for us to understand it, because antibody is one effector mechanism. In the cellular immunity, there is multiple cell types, with each cell having its own effector mechanism, right? So the next shift and the major shift is towards the role of the different cells in conferring immunity in an individual, and also in passing of the immunization state, right? So just a few more names. So we have Merrill Chase. It's in 1940s. So this entire debate was going on since the 1800s, and it was finally settled by the experiment by Merrill Chase, where the immune status was transferred by transferring WBCs in guinea pigs. So guinea pigs who were not infected, they were given WBCs from previously infected guinea pigs. Which pathogen? Transferring WBCs from tuberculosis infected guinea pigs. Right. So from this particular experiment, it was very, very clear that there is a possibility of cell-mediated transfer of immunity. So cellular immunity is very much there. Then the effort started in which cell type, what is the function of everything. So there we had the discovery of lymphocytes. By the next 10 years, lymphocytes were identified. So you have to also understand the technological advances that have happened. So we had a lot of cell culture techniques evolving. Lymphocytes were identified, and so Bruce Glick identified it into T cells and B cells. This was followed by so once we had the knowledge about cells, this was also followed by work on the identification of different antibodies possible, and how this antibody is actually working towards neutralizing the various pathogens that is coming in there. So that is the theory of that is a theory that we will see further in adaptive immune while we are discussing adaptive immunity. But to give a brief, so there is a antigen and antibody interaction. So what is antigen? So far we were saying pathogen, microorganisms, we were naming bacteria and fungi, but the body doesn't know all these names. It just knows that something foreign has come in. It is going to recognize a particular part of this foreign substance. So what is this foreign substance? The body recognizes just one thing, and this one thing is known as the antigen, to give you a simplified version of it. So antigen is nothing but the region which the body identifies as harmful. So against this antigen, it is going to mount we have antibodies coming in. So antibody is already there in the body. How is there? It's an effector mechanism. So certain it's a response that the recognition has triggered, and as a response, multiple antibody has been produced. This antibody is now going to interact with this antigen, and it will neutralize. Okay, it will neutralize the pathogen. So why I got in here is because antibody, again, a very important name, we shouldn't have missed it. Karl Landsteiner Karl Landsteiner showed that antibody could be produced against everything. So it's not just a pathogen or an antigen, or it's not just a microorganism or something which is naturally present. Even if there is a new peptide that I form today and I put it in someone's body, there is going to be an antibody response to it, provided the antigenicity is high enough. So ignore the terms right now, but provided this antigen is strong enough for the body to recognize and put up a response, there is going to be an antibody produced against it, even if it's not naturally present in our environment. Okay, so the next set of questions started. So if there is an antibody that can be produced against everything, how does it, what sort of interaction is facilitating it? How is it possible that such a huge diversity is present? What is guiding this diversity? So therein, so let's just put in words, antibody. So AB is for antibody and AG is for antigen. That's a acronym that you will find throughout immunology lectures. I hope you get used to it. So there is an antibody against everything, and this is very specific. So it can even recognize very subtle differences between the different antigens, right? So how is it able to do it? That was another debate altogether. And from here, two theories were proposed, right? Not by Landsteiner, but in the general. So there's no one person who is given a credit for discussion, right? So there was a lot of scientific research happening throughout, and we had two theories coming in. So theory of selection and instructional. So if you have studied about the enzymes, there were certain models to it, like instructional lock-in key, of which we selected the lock-in key, wherein we have a particular enzyme which is having a particular shape, and whatever is a perfect fitting substrate, only that gets to get catalyzed, right? So over here as well, so instructional theory is somewhat like the induced fit theory from enzymes, if you have studied that subject previously. So what is happening in instructional theory is, if I have an antigen which is, say, round, and I have a antibody which might be in the body, it might be rectangle, having a rectangular fit, this is still going to guide the antibody into a circular fit. So this is instructional, right? Exactly how it happens in the induced fit model, that the antigen is guiding the folding of the antibody. The theory of selection is what picked momentum because of its relevance and what eventually was accepted. So this is the underlying paradigm of modern immunology. So you might pay attention here. So what it says is that there is a variety of receptors on cells of the immune system. Okay. So when these receptors, they can bind an antigen like lock and key. So when these receptors bind an antigen in a lock and key based model, it would induce proliferation. It was induced proliferation of more cells with the same kind of receptor. Right, so this is what came into proposal very early, and this is exactly the theory which was further built on into the clonal selection theory. And it forms the so this is very, very important. So what are the features of clonal selection theory? You might just make a note. So it says that individual lymphocytes. So we was talking about these cells, the cells of the immune system. So lymphocytes in particular, because of this is what was proposed, and clonal selection theory is what is finally modified to perfection and adapted till date. So there's always a possibility of further adaptation, further modification. The scope always open, and that's why we have science and we have research going on despite so much information which is already known to the human beings. So what clonal selection theory is accepted as that individual lymphocytes, they express a lot of receptors on their membranes which is specific to different antigens. So distinct antigens, very specific to distinct antigens, and this specificity is determined before a receptor comes in contact with an antigen. So before the cell even got to the circulating immune system fluids, it knew that this kind of antigen only I can bind to. So the specificity was determined before its encounter to an antigen. When the antigen comes in next, it binds to a compatible receptor which is present on a specific cell, and it activates that cell. So I have an antigen which had the possibility of binding to different cells with different kinds of receptors, but of all the lot, we have a particular cell which perfectly fits to this antigen. This binding of antigen to the receptor on this one particular cell type activates the cell, which causes it to proliferate into cells of the same specific type of receptor. So it causes proliferation into exact identical clone of cells, right? So that is why the word clonal selection is there. So once this antigen binds to a cell of a specific type, it activates it. It causes this particular cell to proliferate into the exact copy of it, exact clone of cells which have the same receptor as the parent cell. So now that is why it is known as clonal selection. So this is the antigen binding which actually selects that which clone of cell is now going to be produced in the body so that the antigen can be effectively neutralized. So that is the clonal selection theory. So, okay. So I think that is all for history component of the immune system. If there is anything that you want to ask or any suggestion that you would like to give for me to carry forward with, if there's anything that is not clear to you, if the network say is not stable, if there's any problem coming up from my side, you can please let me know. Okay, please give me 1 minute. So we will continue. So once we have understood, do we get any notes or recorded lecture after this class? I will be up. We are supposed to share our notes to the portals, and we are also supposed to make a recording of the lectures, which is already ongoing. That is also for information of all. But I'm not sure if it is going to be provided for you for later access. So I'll get back to you maybe in the next class. And if we have a link that is also shared with the student, you will be receiving it from NPTEL itself, right? Okay. Okay, so before we were discussing about the history, we stopped our discussion on the summary of the immune system over here.
Excuse me, ma'am.
Yes, please
Ma'am,
go ahead.
actually, in the same timing, I am having another class as well on the NPTEL. So, will it be possible that I will be getting the recording of it?
See, so I think as far as the live interactions are are to be considered, you actually will have a chance to attend three of the live interactions, and it's your choice to join all of them or choose either of it, right? So if the class timings are clashing, you might also want to join the class, this live interaction of the immunology course on Sunday. I think it's from 3 to 5 p.m. You will be receiving the links every week, or you can also join on the Saturday class. It is from 6 to 8, if I'm not wrong. So every week you will be receiving after the core, after I think 3 4 days into the next week, into every week you will be receiving a mail from NPTEL itself, wherein they give you all the class links of all these three classes. We have different instructors who are guiding you on all these different timings, but as far as the recording part is concerned, I'm not sure if it's going to be shared with the students, right? But you can always join other instructors who are explaining the same things in their own respective ways.
So, ma'am, I will be getting the lectures on the or the live interactions on the immunology by the other faculties.
You can attend their classes live as well.
Okay, ma'am. And ma'am, right now you have talked about the serum, so I think that the RBCs must be absent in that because if we are injecting that into the another non-immunized individual, then if RBCs are present, then they might cause an immune response in that person's body.
If I think, so I'm not sure. It might be that RBCs are not there. It might be RBCs are there. I have to check it once again. But if we think of it like that, I get what you're trying to ask, that there might be certain antigens or certain markers which are present on these RBCs, and if I'm transferring it into the another individual, their body might actually recognize this RBC as a foreign entity and mount a response against it. So it might cause more harm, just how blood transfusion, wrong blood transfusions can cause, right? That is what your logic you're trying to give. But if I think about it like that, even the WBCs, they also have a lot of receptors and markers. So I'm sure that the WBCs are there in the serum component, and think so. But I'll get back to it.
But ma'am, WBC's outer surface receptors must be same in and every individual of that particular species. I think so. Is it
Yes,
correct?
yes. I think you are right. Good. I think you are right. The WBC receptors. So when I talk about T cells, we study about the CD4 and the CD28 molecules. B cells have MHCs, but then MHCs could be different in every individual. So MHC type matching is also happening these days when transplantation of organs are considered.
Ma'am, actually, I am under a second year graduate student, so I'm
Mhm.
not having that much knowledge. But what you're trying to convey is that I'm able to understand. So, is it sufficient for me to know that much, or any advanced level I need to require to understand it?
No, I think it's good that you are trying to apply some logics when I'm trying to ask a question. As I'm saying, I do not recall exactly that whether or not the cells are present in the in the serum component. I'm sure that do not have the clotting factors. As far as what is the cellular components in serum, I will have to check. So I would not really want to give a solid answer that yes, that's the right thing. But it's good that you are trying to reason the possibility of one cell type and the other, of one being not there and the other being present. But I'll surely get back to you in my next lecture. So hope to see you there.
Okay, ma'am. And ma'am, you have wrote antitoxin, agglutination, and precipitation there. So the serum is doing all these functions, or
So what the researchers see, so it was said that serum is having the potential of passing on the state of immunization, right? Now different researchers started to isolate or say identify that what exactly in this serum is having this effector mechanism, what is exactly there to neutralize some pathogenic component, right? So therein they say some toxic substance is there and your serum is neutralizing this toxin. They name that particular substance as antitoxin. So if there is a something injected in the immunized individual and the serum is actually able to precipitate out this particular thing, they say they just name it. So these are just nomenclatures that are given by different scientists in with respect to the particular experiment that they had performed. So based on what is the function that a particular serum component is performing, they just give that name. So it's not like they have so it's just not like that's all there. So as I said, then the story was built up that when the serum was fractionized, so how the fractionization happens, that part you can study. So you can actually fractionization can happen in multiple ways. So you can either centrifuge it and it can be done in a say density dependent gradient manner, and different gradients are there and different gradients will trap in different components of the serum. So when a final study was performed, a particular fraction of the serum was isolated, which had a lot of gamma globulins in it. So every fraction must have been characterized for. So they must have checked that what is the neutralizing potential of every fraction, and they must have found that say that this third fraction has something which is able to neutralize all the antigens. When they further characterized this fraction, they found that there's a lot of gamma globulins in it, which we now know is antibodies that are actually playing a role there. Is that clear?
okay, ma'am.
Okay. Okay. Any questions from anyone else? Okay. So we were saying, so we're done with the history, and now we'll start with the understanding of the innate immune system. Right. So as I was saying, innate is something that is already there in your body since you were born. So your body already knows what to do with it, and so it's always easy to classify things so that things don't get mixed up. So that's why innate immune system is also classified into 4 types of defense barriers. So it's non-specific, right? So what if I'm talking about cells and receptors, those are very specific thing, because receptors is nothing but again certain biomolecules which might be decorated on the cells, and they must be having a certain conformation. Whenever there is a structural conformation or something like that involved, it definitely has some specificity to it, right? But when there is a barrier, say a police traffic barrier, that roadblocks that you might see, it's a barrier and it's common to every type of vehicle coming in. So it's not specific. It doesn't really have a shape that is going to fit into something or anything like that. So there are 4 defense barriers that compromise the innate immune system. Okay, so we'll see each of them separately. The first that comes into play is the physical or anatomical barrier. So when I was giving you an example of the police road barricades, that is exactly what is there in the physical or anatomical barrier. So there is something physically present to stop any pathogen from coming in. Robule. So robule, we are almost towards the last topic of our class today. Hope to see you join on time next time. Okay. So in physical barrier, we have skin, right? Very obvious. The first thing that is covering everything on our body is our skin. So it might look like a very continuous sheath, but we do have certain say pore-like things and structures like it, which is visible also on our skin. But nonetheless, they are still preventing any entry of any superficial pathogen into our internal fluids or internal organs per se. So in skin, what role does it perform? It is keratinized. So keratinization is something. So this is a very obvious kind of topic to understand. So I'll go a little quickly over here. If there's anything that you're not able to follow, please let me know. Right? So skin is keratinized, as in it has a hardened covering to it. So this keratinization provides waterproofing. So that this keratinized topmost layer is exactly the epidermis. Beneath the epidermis, we have dermis. So in the dermal layer, we have a lot of sebaceous glands. So any sort of gland, it's going to produce something. So sebaceous gland produces sebum. What does sebum do? It maintains pH. And what is the pH that it is actually maintaining? I'm not sure, but some of you might not feel really comfortable to know that maybe the girls in the class especially, that the pH that indeed has to be maintained for the skin is around 3 to 5. So there's a lot of cosmetics that actually brand neutral pH products and all those things. So that is not something that is really ideal to the body, right? Why acidic? Because if it will be very neutral, it's going to support the growth of a lot of microorganisms. So acidic pH is one of the barrier against any microorganism that is present, that is settling up or coming in interaction with your skin, right? So the sebaceous gland produce sebum is one of the main factor that is protecting any microorganism from breaching through your skin, right? Apart from skin, we have the mucous membranes. The skin is something that is visible to us superficially, right? But every organ on the inside also has some sort of protective membranes to it, mostly mucus lined membranes. So mucus is a slimy body fluid. So this slimy thing, maybe you can say it helps in trapping the microorganism, because if there's anything sticky, if there is anything sticky, it will not allow it to further cross a particular barrier. And apart from that, so mucous membranes, they definitely have mucus, which we are talking about, which is a sticky thing. And apart from that, there is similarly we have tears, saliva. They all have certain active components which can actually lyse the microorganisms. And again, in this particular, by this particular way, they are able to protect us. So what happens, so in the mucous membrane, say, which is lining our gut, right? So we have the gut-associated mucous membrane. So we'll encounter all of these when we are talking about the lymphoid organs. But just for an example, gut-associated. So if you have studied about the gut linings, you know that there's a lot of cilia lining this entire enteric system, right? And then we have the mucus. So this beating of cilia is actually helping to move out all the microorganism that has been trapped in this sticky slimy mucus. Similarly, we have this in our respiratory tract, in the urinogenital tract, and it all forms a barrier for the entry of microorganisms, right? And so there are two ways. That is what is most important for you to understand. One, the mucous membrane is trapping microorganism, and second, it has some active components that can also lyse and kill the microorganism. Once the organism is trapped or once the dead body is present, it is a beating of the ciliary linings of the different organs that is propelling the debris and the trapped organisms out of your body, right? So this is for the physical or anatomical barrier. Next we have physiological barrier. So this is a little more interesting to understand because this is not something that we usually consider as protective. So what is happening? Every human body has a temperature of, what is the ideal body temperature? See, we love the 37 degrees Celsius roughly, right? Is that true? You might check, right? So let's say everybody has ambient temperature. Every human body has a temperature that it maintains for itself, and there's a reason for it. So at this particular temperature, it sustains all the physiological functions, and it also supports the growth of commensal microorganisms, or organisms that might not cause any disease to us, right? Then we have pH, say the acidic content of the gut, right? The stomach. In the stomach, when the digestion process is happening, you must have come across that the pH of the gut is very, very low, right? So when the pH is low, it is not only just helping your food to get digested, obviously it has a very important role there, but your different microorganisms that might have escaped the membranous tracts of the upper digestive tract, they must have come into the stomach, and that's the second barrier that it encounters, which is the physiological barrier, which is the pH of your internal body organs, right? So this pH is able to it's not allowing the growth of these microorganisms, and they are also going to further die out. They are going to be and then further flushed out. Apart from that, the most important part, we have certain soluble or membrane associated molecules. So I'll just take the names for these because all of these functions are going to be studied in detail in all our following classes, right? What molecules? Insoluble, if I say, just to take some names, we have lysozymes. You must have studied about them in maybe if you have read about cell biology, you might know that in the cell we have certain fluid filled pouches like structure, which is the lysosomes. Within the lysosomes there are certain very potent enzymes called the lysozymes, which can help to lyse the bacterial cell wall, leading to its ultimate death. Apart from that, we have interferons. So what do the interferons do? They are very important for conferring the antiviral state. So what happens that when we have a cell infected by the virus, this poor cell says that I have nothing left that I can do for myself, but let me just warn all the cells surrounding me. So it is going to release a lot of interferons in its surrounding, which is going to instruct the other cells to display an antiviral state. So certain receptors on the nearby cells is going to change because of this particular soluble component which has been produced by your innate immune system, which is not going to allow this viral infection to further propagate, right? Then we have the complement system, one of the most complicated and most difficult to memorize topic that I would find when as a student. And then we have some more names like collectins, PRPs, pattern recognition molecules. So while we started our class, we were saying that innate immune system is not specific, but it definitely gets to recognize some patterns which is specific to different classes of microorganism. Right? So these can be enbriefed as PRPs. Now this PRPs again, so PRPs is something which can be membrane associated also, which can be soluble also, right? So if it is membrane associated, it is called TLRs, toll-like receptors. And if it is soluble, it doesn't have a name, thankfully. So it's just soluble PRPs which are circulating inside your body, and it's going to recognize any sort of pattern which can be associated with any kind of pathogen, and it is going to warn your innate immune system to ensue certain defense mechanisms, certain effector responses, right? So what this pattern recognition molecules are doing, they are going to recognize PAMPs. Pathogen associated molecules. I'm not sure. Just check the full form of PAMPs. It's pathogen associated membrane proteins. It's pathogen associated membrane proteins. Okay? Okay, so that is true for your physiological barrier. Next, so all these things are, it's somewhere related to the barrier. Now we have what is the cellular component of the immune system. So we have phagocytic cells. So this is the, because we like to leave the nomenclature to something like a barrier, because that's more easier for us to relate to for an innate system. It is known as the phagocytic barrier, right? So as it's evident from its name that it comprises of cells. Which cells? Monocytes, neutrophils, and tissue macrophages. So these are the phagocytic cells of the body. So monocytes and neutrophils, and in fact macrophages also, you would find it surveilling through your lymph. So lymph is the, so I'm saying immune fluid, I would not like to, but if you have heard of the name, so we have blood and we also have lymph. So this lymph is actually the carrier of all the WBCs that we are talking about, and it would take it to all the different organs wherein some antigen encounter is possible, and that is where all your immune cells are circulating. So monocyte, neutrophils, and macrophages, they are the surveilling phagocytic cells, and we also have macrophages which are in the peripheral system, but we also have tissue-specific macrophages. So they are all they all together constitute the phagocytic cell barrier. Now, okay, so what is also important?
Excuse me, ma'am.
Yes.
Ma'am, you told right now that lymph is carrying the WBC. So I think that only B and T cells are a part of lymphatic systems, not all, because neutrophils, monocytes, and other rest are traveling in the blood as well, no?
Yes, you might be right. Okay, I can get back with that also. So you're saying that the neutrophils are not present in the lymphatic system?
No, ma'am. Actually, I'm not sure about that they are present in lymphatic system or not, because I think that in lymphatic system the B and T cells maturation and their traveling and cleaning, I think that is taking place in the lymphatic nodes and that all.
Okay, we can check that, right?
Okay, ma'am. And
I
ma'am,
can.
the thing that WBCs are also present in blood, that thing I'm saying in respect to the neutrophils, because they are the first one to come at the infected side. So that's why I'm saying.
See, even your lymphatic system. So no, so yes, you might be right, because the lymph, I think, is only draining the lymphoid organs. So blood is what actually is is going through all your tissue damage sites, say, and it needs to have some phagocytic cells in it to have the effector responses coming in.
Yes, ma'am.
Okay, okay. I'll also look into it, and I'll also get back if there's anything which needs to change for it. But yes, you might be right that the lymph is the only thing that is draining through the lymphoid organs. Good. Okay, should we move ahead?
Yes, ma'am. And ma'am, will you please repeat the topic of pattern recognition molecule, because I don't understand their full forms that you have written in short, TLR, PRPs.
See, TLR's PR, so PRP is nothing. PRP is the short form to the pattern recognition molecules. So what happens? We have the pattern. So it's like a receptor which is present on the membranes, or it might be released as soluble components, right? So okay. Let's say we have a cell which has some molecules to recognize different patterns, or it can either be associated to the cell, or it might be freely circulating in the uh, again, so in the blood or in the lymph. So we have certain soluble factors or certain cell associated factors which can recognize certain patterns. To which all pattern it can recognize. So it can recognize, say, a triangular pattern. So this is just shapes that I'm drawing. But when you're talking in terms of biology, pattern means the conformations that a particular protein is folded to. So it's not like we have an antigen which is triangular in shape. We might have a protein folded together to assume a triangular shape, right? So in this triangular shape, why is it coming together? Because of some pattern of amino acid that is there, which is asking it to fold in a particular shape. Just to keep the things simple, we have a pattern recognition molecule which will recognize a certain pattern which is associated to a certain class of microorganisms. So we have different microorganisms having, because say bacterial cell walls is having rich concentration of your glycans, right? And your fungal cell wall has a lot of chitin in it. So we have pattern recognition molecules which is able to distinguish between these two molecules based on certain patterns, certain amino acids which are there for glycans and certain things which are there for chitins. So those patterns are recognized by the pattern recognition molecules, which can be bound to the cell, which can be soluble, right? So when these factors are going to find one such pattern which is associated on, say, a bigger microorganism, it knows that something foreign has come in, right? So this pattern on the particular pathogen is recognized by the pattern recognition molecules. Now in pattern, so this pattern recognition molecules is known as is just engraved as PRPs. Now further, we can there are different examples of pattern recognition molecules. One such very important pattern recognition molecule is TLRs. So TLRs is again nothing short form to toll-like receptors. So toll receptors, so why it's toll-like? Because toll receptors is something which was initially identified in context of developmental biology in the Drosophila model, because of the certain conformation that particular receptor assumes, right? So that receptor, when it was identified in the vertebrate system, it had a similar look-alike. So people initially thought that it might have a role in the developmental context only. But when they found, when they tried to characterize the role of this particular receptor, so they named it toll-like because of its structural conformation, like the tolls in Drosophila. But it was found that in the vertebrate system, it was functioning as a component of the innate immune system, and it is nothing but a type of pattern recognition molecule. So we have many types of TLRs, which is recognizing different patterns in different kinds of molecules, right? So TLRs is nothing, it's just an example of PRP that you should know as of now. Now we can have, so TLRs are there, and we can also have, so TLR is a cell associated complex, but we can also have many such PRPs which are released in the soluble media, which can again in a similar way function to identify certain patterns on the antigen, right? So what is this PRP exactly recognizing? What is this pattern that I'm saying? So biologists love to name everything. So this pattern is again given a name called pathogen associated. So let me just see if it's, I do not want to write something wrong. Yes, it's pathogen and molecular patterns. So it's nothing new. The pattern that we were discussing so far associated to the pathogen. So the biologists have named it pathogen associated molecular patterns, right?
So, ma'am, PAMs will be found on the antigen surface, and PRPs are present inside the individual body. We can say so.
See, not exactly. PAMP is not found on the antigen, right? When I'm talking about antigen, that is something that is recognized by antibody, right?
No, ma'am, actually, I mean antigen with the pathogen only. Just in the
Yes,
immunology,
yes.
I alternatively use both terms.
Right. So our we'll try to come to the discussion of antigen antibody as soon as possible, because it is very important that which term you are using, because antigen, pathogen, these are very related terms and they might be confusing sometimes. We'll also come across something called as epitope, which is also part of antigen, right? So over here, when we are coming to antigen, that is something that has to be studied in context of adaptive immunity. Here the body has encountered a certain cell, a certain pathogen. The cell can be a pathogen, it might not be a pathogen, right? Again, to identify one as pathogen and identify the other one as non-pathogenic, some patterns associated with this cell is recognized by the immune system. Which component of the immune system? Innate component. The pattern recognition molecules, which is a part of the physiological barriers, recognize these patterns. So these patterns are called as the pathogen associated molecular patterns. So PRP is something which is present in your body, which is the job of which is to recognize the patterns associated with the pathogens. Is this
Okay,
clear
ma'am.
now? Okay,
Yes,
okay,
ma'am.
okay. So is there anyone else who has anything to ask? Okay, okay. So we'll talk about the fourth defense barrier of the innate immune system, the inflammatory response, right? So inflammation in itself is so it's a consequence of four major so whenever there is an infection in your body, there is an inflammatory response. Inflammatory response itself is a consequence of a sequence of events, right? So inflammation is not something that just, so I have a cut and there's an inflammation. That is not how it really happens. It takes some time to get triggered. So how is it happening? So the sequence of events that is actually causing the inflammation is also the four main signs of an inflammation present in your body, right? So inflammation is a sequence of events. So inflammation is a sequence of events, and this sequence of events through its course have four signs, four cardinal signs. So these four cardinal signs mean if you are seeing any of these things there, any of these signs in your body, it is so that you are undergoing a inflammatory response, right? So four cardinal signs. The first being rubor, tuber, calor, and dolor. So rubor stands for redness, tuber for swelling, calor for heat, and dolor for I forgot. We have to see what is dollar for, right? So rubor, tuber, calor, dolor. Any one of you is aware of it? Okay, I'll remember during the course of the lecture, right? So there are three major events that is happening, which is manifesting in four cardinal signs. So whenever there is, so let's just try and understand. I'll also show you a diagram of this. So whenever there is a cut on your skin, so there's an insect bite, right? What is the first response that is there? Okay, so dollar is for pain. The first response that you want to do, you really want to itch that surface because your body tells that there is something that has happened. So there is a sort of irritation there, and it might sometimes also be painful, right? So pain, why that is happening? That is happening as a side, because as a protective mechanism, it is telling your mind throughout that you're supposed to protect the site of infection, right? So that there is no further infection that might happen on your hair, right? Next, as you try to scratch, you see that there is also some redness happening. Yeah, right. Dollar is for pain. So not grief in the context of immunology. Okay. So when you see that there is some, you're scratching the surface, you see that there is some redness that is coming in. Why is that happening? Because there's vasodilation, so there's a lot of blood flow that is happening to the site of infection. Yes, so the blood do have the cells of the, so this is the beauty of science. If you're trying to understand the system logically, you will automatically find answers to your previous questions. Okay, so when there is redness, it is because there's a lot of blood which is flushing through the site of infection. Why is that happening? Because the intention of the body is to bring in these cells which might trigger the effector response. So we do have the cells of the phagocytic barrier in the blood. So Shamika was right. We do have the neutrophils and the phagocytic cells in the blood. Okay. So this redness is happening because of a lot of blood flushing in. Within some time, you will also see some heat. So you will see that if you touch this area, it might be having a slightly warm sort of touch to feel. That is because temperature we read about, right? So at that local site, if there is certain elevation of some temperature, it is also trying to actually take care of any sort of it's an effect of the body to take care or to kill the particular pathogen which might have caused that infection. And in some time, as more and more cells accumulate in this site of infection to take care of it, we might see that if the infection was very serious, there's a lot of cells that have come in. We see that some pus might be also forming, which is having the debris of the these cells, of the pathogen which has been neutralized by these cells, which is all part of this pussy fluid that sometimes oozes off. So all these things is also the accumulation of those things at the site of infection also causes some swelling at time. So these four events which is happening is also manifested as cardinal signs of the inflammatory response. So just to put things in context once again, so what is inflammatory response? We have vasodilation happening, wherein we have the increase in diameter of the blood vessels, because of which a lot of blood can now come towards the affected area, and all the cells that are present in the blood is thereby coming in to the site of infection. In this cell, there is also all the effector cells of the innate immune system, right? So with the so also important, it's not just blood coming in to the site of infection that is going to help, right? There are also these cells which are there in the blood. You must know that infection site might be on the top of your skin, but your blood is present under your dermal layer, right? So it has to, this blood which is carrying all these cells, the cells have to come in at the site of infection, which is the epidermal layer. So for that, the capillaries which is having the blood, they have to now become permeable. They have to allow the movement of these effector cells out of the capillaries into the tissue where the injury is present. So we have vasodilation that brings in blood, which is causing a lot of tissue redness, and now we want the cells which have been carried by these blood to enter the tissue. So the capillaries have to now become permeable. So when they become permeable, we have the phagocytes and the leukocytes which can get into the tissue. Okay, so I'll write that down. So we have vasodilation as a part of the process. So you have vasodilation as the part of the process which brings blood. So this process is also known as erythema. Now we have increased vascular permeability. Okay, why so? So that phagocytic and the lymphocytes can move out. Infection site. Now, so this movement out from the capillaries into the tissue is also not just one thing that just happens, right? It is also a process. So the first part of the process is margination, wherein with the cells of this, so the cell is present through, spread through this capillary volume, but the cell has to now attach to the wall of the capillary. So if we have a capillary which is this thick, we have blood flowing through it, but now the cell has to say come out from this pore. So the first thing it has to do is attach to the wall so that it can now come out, right? So this process of its attachment to the endothelial wall, the capillary wall, the endothelial wall, this process is known as margination. Next, once it has marginalized, it has to move out of this capillary, so which is diapedicis or extravasation. So again, just some terms to what is happening. You need to just, if you follow the sequence, if you follow the obvious sequence of what has to happen, it will be very easy for you to remember these terms. So it has to move out. It gets stuck to the wall of the capillary and now it wants to move out. So when it moves out, this process is known as diapedicis or extravasation, right? Now this tissue is very thick. As I was saying, your skin has a superficial breach. Okay, so it now has to move out. So this moving out process is called extravasation, and your skin is thick The cut is here, the vessels were here, the cell has now just entered the bottom. How would it know that where it has to go? The site of infection, at the site of infection, there is a lot of chemical substances which can actually serve as cues for these cells to reach there. So chemical substances which is being followed, which guides these extravasating cells to reach the site of infection. This process is known as chemotaxis, because taxis is movement and it is moving across a chemical gradient to reach the site of infection. The movement overall is known as chemotaxis, right? How the name suggests. So if you keep breaking up the names, you would know exactly what is happening in the process of it. Now, once it has reached the site of infection, it would release some lytic substances. It would release the lytic substances and the pathogen is going to be killed. Okay. Okay, let me just see if I'm missing on anything. Okay, so yeah, so far this is good. Now there are certain molecules and certain chemical mediators which is actually helping this process to happen, right? So this is the process of inflammation, but we have certain players which is facilitating this process of inflammation. So what is it? Let me just put it here. So we saw about vasodilation. Vasodilation and increased vascular permeability is taken care of by histamine, which is known as one of the primary mediator of inflammatory response. So what it does, histamine can bind to the nearby receptors present at the site of inflammation, and it would cause the vessel to get dilated in order to allow more blood flow, and it is also going to increase vascular permeability. Right, so both the aspects is taken care of by histamine. Then we have, so as I was saying, these chemical cues, chemotaxis. What is there? We have some proteins called as acute. So I'm just giving you some names which we are going to get into details while we are understanding each process in its exclusivity, right? So we have certain things called acute phase proteins. An example of which you might have heard, which is the C-reactive protein. So why is it called C-reactive? Because of the certain component of the pathogenic cell wall that it recognizes, it has been named so. So for now, just remember the examples that I'm giving you. How when these things come up in the following subsequent lectures, we will discuss everything in details, right? So this is an overview of the innate system that we are studying right now. Apart from that, we have so other mediators which can trigger the inflammatory response. So say this sequence of events, what can trigger is, so one such player is also bradykinin. So one of the function of bradykinin is to stimulate pain. So pain, so that you protect your site of injury. Right? So these are certain mediators which is important in the context of inflammatory response. I think with this we can stop. The time is already up for me. I'll just see if there's anything else. Okay, so this we can stop. With this, we finish our discussion on innate immune system. And there's some diagrams that I would like to show you, just that will also help us summarize what all we have learned in today's class. And in this process, if there's anything that any one of you want to discuss, or you want to ask me some question, if any part of today's lecture is not clear, you can please get back to me. So so this is the chapter that I'm referring to in QB. It's the first chapter. This part is the historical perspective. This is a very nice table wherein you will also find a lot of other names that we might not have touched today because of all the constraints with time. We have discussed the theory of clonal selection, and then we have moved on to innate immunity. So QB is a very systematic book, as you might be seeing. So we have a lot of summary tables very nicely explaining everything in a quick manner, and it is very good to follow for a quick overview, right? So this is all the innate mechanisms that we have discussed. We have discussed about skin, mucous membranes, the physiological barriers, phagocytic, endocytic barriers, and the inflammatory barriers, right? Apart from that, so this part is all the different components that we have discussed in details. Yeah, so this is a very nice image. So this is, I'm not sure if it's a cartoon graph or if it's something that has been. So this is the actual electron micrograph of what might be happening when has the screen changed for you? Do you see a black and white image? Yeah, right. We now see it. So this is an electron micrograph, and which you might be seeing. So these structures are the bacteria, and this is the endothelial lining of the urinogenital tract, I suppose. Yeah, urinary tract. So where the bacteria is adhering to. If we now see, so this is a monocyte, a phagocyte, which has trapped a lot of E. coli. So this is a RBC, a red blood cell, which is much smaller in size. So this is a monocyte and a macrophage. So macrophage has a lot of projections. Monocyte might be facilitating the activity of this macrophage to trap in all the different bacterial cells that it has encountered, right? So once it has encountered the bacterial cell, the next job for it is to phagocytose it. So the process of phagocytosis can be broken up into, again, a multi-step process, which is very easy to understand. We have a cell, we have this cell. These are the pseudopodias of the cell, right? And we have this bacteria. Now the pseudopodial extensions are going to close around the bacteria. So it comes inside of a phagosome. This phagosome, this is the lysosome that we were talking about. So this lysosome has a lot of lytic contents, which is shown as black dots in it. So these two are now going to fuse together into a phagolysosome. So the lytic contents are now getting released inside of this phagolysosome, which is going to inside this phagolysosome, which is going to break up the bacteria, and it is finally processed out from the cell, right? So this is how a bacteria can be cleared off by the phagocytic cells. Okay, so this is the last thing that we have discussed today, the process of inflammation. So we have the dermis. So this is the skin, the epidermis, and the dermis. Behind in the dermis, this is all the glands that we were talking about, right? So we have a tissue damage. This tissue damage, maybe a sharp piercing object. So this piercing object also got along with it certain bacteria. This bacteria is now released. So is now released inside this tissue. Behind it, here we have the capillaries. The capillaries have a lot of cells, the RBCs and the phagocytic cells, which we were confused about that whether or not they're present in the lymph or in the blood. But see, here we have the phagocytic cells in the capillaries, in the blood. So the cells first in step 1, as the blood is flowing through, the cell gets margination. Now it has to next move out of the capillary to reach the site of infection. So this process we discussed at extravasation. How it is reaching this site? Via chemotaxis. So this cut over here is producing some some components. So okay, not this. It is producing certain factors here, which is forming a chemical cue for the cells to reach the site of injury. Along with it, the along inside the blood, so this part is the soluble factors that is present in the blood that we have been discussing. So the soluble factors also come out of the capillary because of increased vascular permeability, and along with the phagocytic cells, these components are also taking care of this site of injury, right? So that to neutralize all these pathogens shown in red color. Okay, so
Excuse me, ma'am.
yes.
Ma'am, the tissues are showing the metastasis property due to which they are intact or in close contact with each other. So, how this macrophage is coming there and leading to the site of infection?
But the tissue doesn't show metastasis. What do you mean by? So metastasis is what? When we have a cancerous overgrowth, and when the cancer is firstly overgrowing, it is overgrowing. It is first growing out in as a closed ball, say. Now, when one cancerous cell breaks off, reaches some other site to seed another growth there. So this process is known as metastasis, which is a case for in the case of cancerous cells. Tissue shows metastasis, I do not understand.
Ma'am, I mean that tissues are in cells are in close contact with each other. So, how they are permitting this the cells of the immune system to get there to the site of infection?
There is always an extracellular matrix. So it's
Okay,
not
yes,
like
ma'am, I got that.
so it's not like this particular cell has to move through the path of the cell body, another cell's body, right? There is always extracellular matrix which is cementing all the cells together. So that matrix, the spaces within the matrix which is enough to accommodate these cells to move it, right? So that is how the phagocytic cells or the soluble factors and other things are reaching the site of injury.
Okay, ma'am. And ma'am, one more that you have told that about PRPs. So are they are able to modify themselves according to the foreign substance that they are coming in contact, or they are having their specific structure only?
See, as far as I think, they do have their specific structure, but that is something I will have to look back to be sure about. But I think they do have a specific. Uh, so it's like they are not very choosy about the structural detail of the PAMPs, right? So even closely resembling patterns can be recognized by a particular PRP, but whether or not the PRPs are flexible in their own conformation, or so it's not like one PRP is going to recognize just one type of PAM, which is true for antigen antibody interaction, which is very specific. So one PRP might not be very picky and can identify similar or similar appearing PAMs, right? It might not be the case that it will only bind to say the sequence of ATG, if you're talking about your nuclear pattern. So nuclear patterns cannot be true for PAM. So I think you get the point that if I have a protein and we have 3 amino acids, maybe if one is changed, this PRP can still bind this PAM region, right? It is not very picky. Whereas for antigen antibody reactions, even if there is one amino acid change, the antibody will not be binding to the particular antigen. There might be some other antibody which is specific to that sequence, which can now come and bind to it. But PAMP and PRP interaction is not that specific. But whether or not the PRPs are flexible once they are present on a cell or in a soluble component, that I have to check. So I'm sure
Okay,
it
ma'am.
is not going to be like a induce fit kind of a thing. But how far is
Yes, ma'am.
the it will not be an induce fit kind of a thing. They're
Okay,
not
ma'am. So it is
specific.
not the thing that we see in the case of enzyme like lock and key
No,
hypothesis. It is
no,
totally different, right?
no. Yes. It is not
Okay.
like a lock-in key mechanism. It's not, it might not want a perfect fit, but will definitely want something similar to what it is able to recognize. A perfect fit, if it's not there, it might still just find. A perfect fit is needed for your antigen antibody interaction to happen.
Okay, ma'am.
Okay, so in the next class, when we take up adaptive immune system, we will first again discuss all the features specific to the adaptive immune system, and then we will get into the details of all the antibody and antibody, what is antigen, the different types of it. Just we'll cover as much as the time permits, right? Is there anything? So I think the cell differentiation part has already been taken up by Professor Ghosh in the week one of the lecture. I wanted to keep it a little slow for today so that I know how much all of you are able to follow. So I think we might just do it in the next class. That's not a problem. It's just a diagram that you might have want to understand. But apart from that, from week one, I think we have taken up every other thing, right? Is there anything that I missed that you would want to add, or is there any other discussion? So the assignments that you have to solve, if there is any doubts from the assignments that you have received last week, for all that's true for all our following classes. You might just want to discuss the assignments that you have received, and if there's any doubt, we can take up some of those questions too. In fact, all of those questions too. Okay. So I'll just give you 2-3 minutes more if any one of you want to revisit something, or any one of you can think of another question, or within that time, I'll then close the session.
Excuse me, ma'am.
Please go ahead.
Ma'am, actually, I have joined this course late 1 week, so I have to
Mm-hmm.
do my prior assignment as well.
Mm-hmm. So your week zero assignment, you mean?
No, ma'am, week one, I think.
So this discussion which we right now had, beta, it's for your week one. So the thing is, since I'm taking a Tuesday classes, I am going to actually still discuss whatever you have done in your previous week. So today's discussion is for your previous week. So if you, I think you guys still have the assignment there, and you have to submit it on time. I'm not sure. I can check that. That is there a stipulated time? Maybe your co-learners can help with that question. Is there a stipulated time within which the assignments have to be submitted? Maybe
Yes,
that.
ma'am,
Yes,
I
ma'am, I
think
think
that
that
it is
it is
there.
there. Okay, okay. So if your portal doesn't allow you to submit that previous week's assignment already, because you must have run out of the time, then I don't think there's anything that I can help with. But for your information, you can always go through those questions and try to solve them on your own, so that you know how much where you stand in terms of your learning. But as far as evaluation of that assignment for your course credits, I don't think that is going to be possible.
So, ma'am, the questions will, or the assignments question will be available to me.
Are you not able to find them on the portal?
Ma'am, right now I have not checked so.
Please try to check that, because when I try to do it, I do see the previous week assignments there. So I'm think that you all might be having an access to all the assignments, despite of the fact if you submit them or you not, or you do not submit them. I think if you're also registering for your exam, then you need to submit the assignments, because the some marks in the assignments is going to be
Yes,
considered for
ma'am.
evaluation.
Evaluation purpose they will be considered.
Yes, yes. So for that purpose, you will have to actually submit the assignments on time. But as far as for your knowledge, you want to just see and solve, I think you will be able to view the assignments for sure. And if it's not visible to you, as it is visible to me. So if it's not visible to you, maybe I can share a PDF in some resource links if they allow us to like provide one. So I can otherwise create a drive link, and I can put up assignments, and I might share that link to you in the next class in the chat box.
Okay,
Just try
thank
to
you so much,
find
ma'am.
out once if you are able to see that assignment. I think you will be able to find it.
Okay, ma'am. And ma'am, how we can connect with you regarding today's class doubt or any other doubt that we have come across?
Have you guys been given? So I will be coming here every Tuesday, 6 to 8. So that the best way for you is to always join the sessions if you want to take doubts with me. Any class, any previous class, anyway, it's possible. But in specific, if you have a doubt, you can also put it on the discussion forums which have been created by NPTL. You must be having an access to it.
Yes, ma'am, I am having that access to that portal.
So you can
But
always post. So we, the educators, can also see all the questions that you all are putting up. So if particularly you want a doubt to be taken up by me, it would be very helpful if you can please mention that. Maybe Debalina ma'am, can you please solve this? So that if I see my name, I'll definitely get to it. So I do check that forum every week before coming to the class, so that I know that if there's a particular doubt which is common to some other students, the others might have a problem with it.
so,
Like
ma'am,
if
I need to mention your name in the discussion forum
See,
while I.
I check almost all of them, right? And if I have a doubt which has already been answered by someone, or if there's a long discussion which is going on amongst the student, I would not like to disrupt it with my opinion, because when the students discuss, there's a lot of things and ideas that this exchange, and it is the so discussion is the best form of learning that I feel, right? But if you specifically really want me to address a question or something like that, it would be very helpful if you can mention my name there. So then I'll definitely get back to it, right? So you can post your queries there, and you can always bring your queries to the next class. Okay,
Okay,
and
ma'am.
I will always encourage all of you to try and interact with all the scholars from the live session. So we are all PhD students from different institutes who are taking up the live sessions. So that will also help you to see that how far you are comfortable, the speed and the way of explaining, and all the things that tend to vary from one person to other. So you might want to also check the live session, all the 3 live session, for you to find the best fit. Okay, so that's for like all of you. Okay, should I stop the sharing? Okay, so if there's no further doubts, I can close the session if that's okay. Okay, I'll hopefully see you next week, right?
Thank you so much, ma'am.
Okay, thank you. And it's good that you interact. I really, really encourage Shamika for asking up everything that she's not clear with. So please, it's a platform. All of you are equally part of this platform. So the purpose of this interaction is to, in fact, keep it as a doubt clearing session and not a session where we have to teach you, because you're already getting all the recorded lectures. But since students tend to not interact, we have also been told that you can like revisit all the concepts that have been already taught for the week, so that anyone who has a doubt can like come up with it. But if we keep it interactive, we can also like shorten our lectures and keep the interaction on our priority. So that's always the first priority. This is like an interaction for your doubts and queries and things like that. So I really, really encourage all of you to be as participating as Shamika was today, right? Okay. So I'll take a leave. I hope to see your names next week. Okay, okay. Good night.